Okay, welcome back. This is our last section in terms of chemical basis of life to organic molecules. And we're going to finish up our discussion of organic polymers looking at the nucleic acids. So let's get right to it. There are two types of nucleic acids, DNA and RNA. And so what's going to be important here is that you understand the relationship between structure and function. Now for RNA, we're going to come back and look at it in more detail structurally when we talk about transcription. And for now, we're just going to do the basics of the structure and function of DNA. So most people are familiar with the structure of DNA. It is this classic double helix with two strands of polymerized nucleotides that are wrapped around one another. And its main function is in storage, and that seems a little bit strange because we talked about storage before in terms of energy storage or carbon storage when we've been looking at lipids and carbohydrates. The storage we're talking about here is information. Now, when we get to talking about transcription and translation, that's gene expression collectively, it's going to be very important that we talk about the storage of information and the flow of genetic information. So DNA stores genetic information in the sequence of nucleotides that make up the two strands. And the fact there are two strands is going to become important when we talk about DNA replication. So what else does DNA do? Well, DNA has to be responsible for inheritance. That means it's got to be responsible for passing characteristics from parent to offspring, to their offspring, and to their offspring. And so when we say inheritance, that's what we mean. It also is important that it can pass information from cell to cell during division and in horizontal gene transfer, which we talked about a couple of weeks ago now. And so you can see here we've got the Kennedy brothers, and they all look pretty similar. They're a great example of characteristics being inherited and how the three sons look somewhat like their parents. If you look at Hillary Clinton and her daughter, they look very similar as well. So DNA's structure has to account for its ability to be inherited. And this comes down to the nature of DNA replication, which we will get to when we talk about DNA replication in more detail. So what are the structures, what are the functions do we have? DNA's got to be responsible for the similarities and the differences that we see between individuals and between species. So for example, that's me on the left there. And if you looked at me and you looked at my mother and father, you would see there are some similarities. And if you compare me to the handsome fellow on the right here, Pan troglodytes, the chimpanzee, you can see some similarities there as well. Hopefully not too many, but you can certainly see some similarities. So the structure of DNA and the sequence of the nucleotides in the molecule has to account for the similarities that we see between related species, between related individuals, but it must also account for the differences that we see. So if we compare two distantly related species or two distantly related individuals, we would see more genetic differences. Those are differences in the sequences of nucleotides in the DNA than what we would if we compared two closely related species or closely related individuals. So if we compared me and my mother, we would see more genetic similarities than if we compared my mother with, let's say, her grandson. There would be fewer similarities between the grandson and my mother than between me and my mother. If we compared the genomes of me with this handsome chimpanzee on the right-hand side, we would see more similarities in terms of the sequences of nucleotides and the structure of the genome, for that matter, than if we compared me and a pufferfish, for example. There'd be some similarities, but there would be fewer similarities than there are between me and the chimpanzee. So the structure of DNA and the sequences of nucleotides in that molecule have to account for the similarities and the differences we see between related and unrelated individuals. Now we're going to go back to our theme of looking at the monomers that are used to build these organic polymers. And so nucleic acids, whether we're talking about DNA or RNA, are assembled from nucleotides. And so the nucleotides are joined together into strands, and those two strands in the case of DNA wrap around one another with the complementary bases, those are the matching bases, facing one another, joined together by hydrogen bonds. That gives kind of like the rungs in a ladder structure that you see in molecules of DNA that are modeled. So let's take a look at a nucleotide. Here's a typical nucleotide. So nucleotides have three parts. Here's the first part here. That's a nitrogenous base. It's called a nitrogenous base because it's got some nitrogen in it. So there's a nitrogenous base, and the nitrogenous base is attached to a 5-carbon sugar. Now, we talked about carbohydrates when we talked about hexoses. Those are 6-carbon sugars, but this sugar here has 1 carbon, 2, 3, 4, 5 carbons. So this makes it a pentose sugar. So we've got the nitrogenous base attached to the pentose, and then we've got attached to the pentose sugar, we've got at least one phosphate group. So some nucleotides will have 2, some nucleotides will have 3. When we look at the structure of the nucleotides in a DNA molecule, you'll see that there is one phosphate molecule between adjacent nucleotides. Now with the nucleotides, there's some important chemistry that we can't get away from. So when we look at the base, it's attached to a very specific carbon in the pentose sugar, and that carbon there is called the 1' carbon. Now it's called 1', it's given the prime designation because these carbons and nitrogens up here in the base are numbered 1 through however many atoms there are there. And so you can't call this the 1 carbon because it's easily confused with the carbons and the nitrogens in the base. So it's called the 1' carbon. So the base is always attached to the 1' carbon, and then there's a 2' carbon, a 3' carbon, and the important thing about a 3' carbon is that it always has this hydroxyl group here, the OH group. Then we've got the 4' carbon and then the 5' carbon here, and we've got attached to that at least one phosphate group, which we've met before. This one's charged and it's got a little hydroxyl group there too. So it's important that you know which parts of each group are linked to other parts of the molecule. So the base is attached to the 1', the 2' here, we'll come to in a moment, is going to be important. 3' has a hydroxyl, 4' has the phosphate group, 5' has a phosphate group attached to it, and then we have the phosphate group. So let's take a look at these pentose sugars. The pentose sugars are important. A lot of people focus on the bases. Those are the nitrogenous bases: adenine, thymine, cytosine, guanine, and uracil in RNA. So 5 bases, but you only find 4 in DNA and 4 in RNA. And a lot of people focus on the bases, and we're not going to do that. We're going to focus on these pentose sugars, and we'll come back to the bases in a moment. So here are the 2 types of sugars that you will find in nucleotides. The one on the left is called deoxyribose. Deoxyribose. So this is deoxyribose, and the one on the right is ribose. Now, if you look at them carefully, they're identical, except for the 2' carbon. So here's the 2' carbon of deoxyribose, and here is the 2' carbon of ribose. So over here on the deoxyribose is what you see is there's a single hydrogen. But over on the ribose, there's a hydroxyl, and the hydroxyl there contains an oxygen, obviously. So the very reason this is called deoxyribose is because it lacks an oxygen down here. So if you've got 2 pieces of nucleic acid, and you've got no other way of telling them apart, if one's DNA and one's RNA, you have to look at the pentose sugars, and you can then tell the DNA from the RNA, because the DNA is deoxyribose nucleic acid, and it doesn't have a hydroxyl group on this 2'. If it's an RNA nucleotide in an RNA molecule, then we're going to have a 2' hydroxyl. That's the only way you can tell DNA and RNA apart if you do not have uracil in the piece of DNA, in the piece of nucleic acid. So if you've got a piece of nucleic acid and it contains uracil, then it's RNA. If it contains thymine, then it's DNA. But let's say you've got a piece of nucleic acid and it's got adenine, cytosine, and guanine in it. You've got no way of telling whether that's RNA or DNA unless you look at the pentose sugar. Now, as soon as we throw uracil or thymine in, you can make some conclusions based on that. So the pentose sugars are very important. Okay, let's take a look at how these nucleotides are linked together. So let's take 2 nucleotides. Now these are drawn a little bit differently, and you've got to get familiar and comfortable with looking at molecules drawn differently. So these are drawn a little bit differently. They're just a little bit rotated, basically. So here's the nitrogenous base of one nucleotide. Okay. here is the pentose sugar, and here is the phosphate group. And don't worry what's up the page, we'll come back to that in a moment. And then here is the second nucleotide down here. Here is the nitrogenous base. Here is the pentose sugar. And now the phosphate group is down here. So that's of the next nucleotide down. So here's the phosphate group that belongs with this nucleotide. So this is a nucleotide at the top here. And then this is a nucleotide here. And then this is the phosphate of the next one down. So these nucleotides have been joined together. So what we do is we take the hydroxyl on the 3' carbon of one nucleotide, and we do that condensation chemistry, and we combine it with a hydrogen atom in a hydroxyl group that was on the phosphate group, and that hydrogen and the hydroxyl from the other nucleotide gives us a water molecule, which we saw last time. Now, I don't worry too much about the chemistry with this, because it's a little bit more complicated and more difficult to draw out. So, because what is important here is that the bond that's resulted from the condensation chemistry that contains this phosphate is called a phosphodiester bond. So if you think back when we were talking about carbohydrates, we talked about the monomers, the chemistry to join them, the chemistry to break them apart, the type of bond that gets formed, and then the structure and function business. And we've been talking about structure and function a little bit. The bond that joins two nucleotides together is called a phosphodiester bond. So that's important to know. So we get these chains of nucleotides, and when two of those wrap around each other, we get the DNA double helix. Now, RNA is single-stranded. That's not to say you can't get regions that are actually kind of folded back on one another and look to be double-stranded, but it's actually only ever one single strand, whereas DNA is double-stranded. RNA can have the appearance of being single-stranded because pieces can fold back on one another, and we'll look at that when we look at the structures of ribosomes and other types of RNA that when we talk about gene expression. So that's pretty much the basics of what we're going to deal with here in terms of structure and function of nucleic acids. Let's finish up like we usually do with some questions. Here they come. Let's see how you get on. Here comes the second one. Now, I've actually misspoken a little bit. I do have an extra couple of slides of some important stuff we've got to talk about, so just bear with me a couple of moments. I didn't do that intentionally, I just forgot I had an extra couple of slides that we need to talk about. So, let's finish up with looking at some base pairing rules, and you're probably familiar with the fact that A base pairs with T, adenine base pairs with thymine, C base pairs with G, cytosine base pairs with guanine. And so if you have an adenine in a piece of DNA, you must have a thymine in a piece of DNA. And if you have a cytosine, you must have a guanine. So that means the number of A's equals the number of T's in a piece of double-stranded DNA. If you have a double-stranded piece of DNA and you know the number of C's, you can work out the number of G's. And then obviously, the total number of nucleotides is the A's plus the T's plus the C's plus the G's. That's going to add up to 100%. But then there are some interesting derivations that we can come up with based on that information. So keep in mind that A equals T, C equals G, and A plus T plus C plus G is 100%. So if you know that a double-stranded piece of DNA is 10% adenine, it's got to be 10% thymine as well, yeah? So 10% plus 10% is 20%. That means the rest must be C plus G. And because C equals G, then we know that whatever 100% minus our 20% for A and T, which is 80%, divided by 2 must be the proportion of C and the proportion of G. Now that's a little bit complicated to follow through, so I've got a couple of questions now which will help you think about this. You're going to have to think about it. So let's see how you get on with seriously, these are the last couple of questions, and then we'll be done with this complete module on organic polymers. So a couple of questions, let's see how you do. Okay, here's the second question. And just before we go, don't forget, RNA is single-stranded. People always forget that RNA is single-stranded, and it's got that ribose sugar, not the deoxyribose. So that is the end of our module on organic polymers. We talked about proteins, we talked about carbohydrates, lipids, nucleic acids. And is what you're going to focus on here is the basic monomers, the chemistry to join them, the chemistry to break them, the bonds that form when you link the monomers into polymers, and the structure-function relationship. So use that to guide your studying, and we'll be back next time and we'll start a discussion of membrane structure and function.